I like to start by saying that since the discovery of X-rays more than 100 years ago, every time when we talk about the biological effects of radiations, one way or the other, we always have linked it to the nucleus of the cells, and specifically to the DNA. So when we talk about DNA damage, it will cause cell death. When we said that if the DNA damage is not repaired, we will have a mutation. And if the mutation is not repaired properly, and if they accumulate, we will result in cancer. All right? And so as a result, when we try to estimate the radiation risk, particularly regarding the late effects of carcinogenesis, as a functions of radiation dose. And traditionally, we have used so-called these linear no threshold. So based on the survivors in the atomic bombs in Hiroshima and Nagasaki, we know where the bomb drops, we know the epicenter, and what is the distance from the epicenter. We could calculate the dose. And so we will be able to have a very precise enumerations of the cancer risk as a functions of radiation dose measured by the distance from the epicenter. So this is a strict lines. In essence, this is all the data we have. Okay, but quite frequently, when we try to estimate the dose below this critical range, which is about 0.5 millisievert or sievert, we don't really have any estimate. And so all these are based on projections of risk, so-called the linear no threshold, in the sense that we will draw strict lines to the point of origin where there's absolutely no dose radiation, which is impossible because every time we breathe, we are exposed to radiations one way or the others. Okay, but nonetheless, this is what we have. And so these linear no threshold dose is based on the assumption that the nucleus is the point where we do the calculations and come up with the numbers. Okay, and so for a number of years, a lot of interest has been discussed on the biological effects of cytoplasmic irradiations. What is happening to the cells if we focus only traversal of the cytosol without touching the nucleus? So in the old days, there are studies where investigators will put a little plutonium tips right above the cells and try to avoid the nucleus and just expose the cytosol. Needless to say, this way of doing things is not precise. We do not know the absolute dose, and we certainly cannot rule out the possibility of straight nuclear irradiations until the developments of a microbeam. Okay, there are various microbeams around, and at the Columbia you have a little tour already. You know the charged particle microbeams that we have, and in essence based on a synchrotrons that you have already have the tours. And from these through a series of collimators, we have these exposures into the target area where we plated the cells, and the cells will be imaged, and automatically the image analysis systems will be able to locate and irradiate the cells one at a time. Da-da, we finish one. Da-da, we finish the second. All right. So the time is approximately about a second to locate and irradiate, and the precision is down to 1 micron. So how small is 1 micron? In theory, you could isolate it and irradiate a single mitochondrion. Okay, so we will come to describe some of these biological effects later on. So about a decade ago, I have a student, a PhD student from the Chinese Academy of Science. He came to Columbia University, and I was giving him a PhD thesis project. And I said, young Dr. Wu, let's go and find out if we were to use a microbeam and we only selectively irradiate the cytoplasm without touching the nucleus of the cells, what will be the biological consequence? And prior to that, we do not know. It's a black box. So using the systems that we have over here, these are the imaged cells. These are the human hamster hybrid cells, and we will describe to you in a minute, in a sense that he will place a particle over here, 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 and selectively avoid targeting the nucleus. He collects all of these cells, and he look at both survival as well as mutation as an endpoint. Okay, and turns out that if you were to collect the cells, analyze the mutations, these are the number of particle traversal through the cytoplasms. These are the mutations in the nucleus of the same hit cells. So we target the cytosol, but we actually measure the mutation in the nucleus of the same hit cells. All right, so the difference is that if we were to look at the yield, these are the mutation, it go up and then eventually level off. So with the 8 particle traversal, we roughly get about 120 mutants, and these are the induced mutants. That means the background has been subtracted. All right, so if we were to compare this yield with that of a direct nuclear traversal, then these numbers will have gone up 4-fold, up to the roof. So direct nuclear traversal is still much more efficient in terms of mutant inductions. All right, and but nonetheless, if you were to target the cytosol, you still be able to induce the mutations at a lowered yield. But one of the important things is that if we were to pluck out the mutants, okay, and look at the types of mutations that we are generating, it tells a very different story, and actually a little implication on the mechanism of how this derive. So the cell system that the work that was published in 1999 was a cell system which we thought and we believe is the state of the art in mutation assay systems. And so this is a CHO, Chinese hamster cells, with a single copy of human chromosome 11. And so these human chromosome 11 on the short arm, we have various marker genes, and this is the centromeres. On the 11p13, we have the CD59 markers, and these genes code for specific cell surface marker on the cells. So if we were to use antibodies against the CD59 in the presence of complement, wild-type cells that controls the genes, and with the genes, therefore the surface markers, and when we target them with antibodies and complements, the cells will be lysed. Okay, if there's a mutations and the markers lost, and when we challenge them, nothing will happen. The cells will proliferate happily. They will form colonies, and that's how we score the mutations. But on the other hand, on the short arm and the long arm, we have a bunch of other markers that we will be able to quantify to define what are the types of mutants that we generate. And so if we were to do that, these are the results that we have. Okay, remember the markers that we use is the CD59. At that time, it was also called the M1C1 genes, 11p13. All right, and so if you were to look at a bunch of spontaneous mutants, every single line is representative of a single mutant, and most of the spontaneous mutants, almost all of them, derive from the loss of just these markers. And if you were to look at those that are induced by cytoplasmic irradiations, it's actually very similar to that of the spontaneous mutations. All right. On the other hand, if we were to look at the type of mutants that are induced through the nuclear traversal, and remember every single line is a single mutant, and most of these derive as a result of marker loss, which suggests that these derive as a result of chromosomal loss, so-called multi-locus deletion. Okay, so many of these that are induced through the nucleus derive as a result of chromosomal loss, which is consistent with what we know about chromosomes, about radiations, which is a very efficient clastogens. Okay, and so with these, then if you were to look at the spontaneous mutants, most of these spontaneous mutants arise as a result of oxidative stress. And so this is the conventional wisdom. So background mutation arise as a result of oxidant stress. And so we wanted to ask, what is the role of reactive radical species in the reductions of mutations among those that are traversed through the cytosol? And so one of the approach that has been used by many laboratories is the use of a fluorogenic probe. Okay, these are molecular chemicals that when they diffuse into the cells in the presence of free radicals, they will convert them from a non-fluorescent forms to a fluorescent forms. And the most common one that we use is so-called the chloromethyl dichlorophysin diacetate. Okay, DCFDA. And so when these probes get into the cells, these are all human fibroblasts, and in the absence of free radicals, you have a little background incidence, but in those that are traversed through the cytosol, you have a much higher intensity of the staining. Okay, so these suggest that when we irradiate cells through the cytosol, we actually produce free radicals. Okay, but this is only one of many confirmational studies that you have to do in order to confirm that these is indeed mediated through reactive radical species. And so one of the approach that we could use is that if free radicals are important in the mutation yield induced by cytoplasmic irradiation, what happens if we remove all these free radicals? Okay, and so by using a radical scavenger such as dimethyl sulfoxide, DMSO. Okay, so these are the 4-alpha particles. These are the traverse through the cytosol, induce the mutations. And if we were to add a free radical scavenger, so-called a quencher, these can then reduce the results in the reductions of the mutation yield. And so these provided a second line of evidence that the biological consequence of targeted cytoplasmic irradiation is indeed mediated by reactive radical species. Okay, so alternatively, your cells and my cells, we all contains ample supply of antioxidants, right? So the MSOs, and you have glutathiones, and these offer some kind of protections of our bodies against free radical damage. And one of those that has been used is glutathiones. And so one of the inhibitors of glutathione synthesis is the so-called bethonium SR sulfoxamines, this BSO. And this BSO, if you were to treat it to cells and tissues, they can inhibit the formations of glutathiones. So in the presence of BSO, your intracellular stress level will increase. And therefore, if you were to irradiate them at the same time with a nuclear, with a cytoplasmic dose of radiations, these can then result in a synergistic response in mutation yield. And so these provided a third line of confirmational evidence that in that case, if you have a depleted intracellular oxidants, you will enhance the biological effects of targeted cytoplasmic irradiations. So over the past 10 years, a lot of things has been shown with what cytoplasmic irradiation actually can do. And so these are work through mainly a series of studies conducted by a number of postdoctoral fellows in my laboratories. And Xuan, who was in the lab for a number of years, now she's a professor, so is Mei Hong. So some of these works are cited over here. But if we were to target the cytosol, we could induce the productions of radical species, third through the inductions of superoxide anions, and gradually resulted in the formations of hydroxyl radicals. We further shown that targeted cytoplasmic irradiation results in the productions of reactive radical species, and these resulted in the inductions of mutations. And if free radicals is indeed generated, they should result in the inductions of oxidative damage, such as 8-hydroxydeoxyguanosines. All right, and we will show you some evidence that lipid peroxidations are happening, and therefore these can also promote the inductions of other radical species. In addition to reactive oxygens, there's also the presence of nitric oxides that you have heard from Professor Matsumoto here this morning. And these, in the presence of superoxides, can generate peroxynitride anions and can therefore further damage the membrane through lipid peroxidation process. All right? So everything that is on this chart has been documented and been published, and it provided a very clear-cut evidence of how targeted cytoplasmic irradiation results in the productions of radical species. All right? Any questions so far? So we have to make sure that everything we have discussed you understand. If there's anything that is not clear, just let me know, and we would go back and make sure we have 100% customer satisfaction. Yes, please.
OHDG is in the nucleus, or you're talking in the cytoplasm?
The OHDG is always in the nucleus because
I thought that hydroxyl radicals cannot move very far. They can go very, very small distance.
That's right.
So how do they cross the nucleus?
That's a very good question. So if you have a chain reaction, okay, and if the site is closer to where the point of damage can occur, then it could damage the nuclear membrane, or it could damage any membrane structure, such as endoplasmic reticulums, and these can propagate the process. And we will also provide you a little evidence later on that these could also result in mitochondrial damage. And if mitochondrial damage is generated, then there will be a constant source of reactive radical species that can produce, that basically form a chain reaction. But you're absolutely right. Many of these free radicals are short-lived, and they have to be produced right on the site where the damage is supposed to occur. All right?
They get past the nuclear membrane.
The process through the nuclear membranes is one of those issues, one of those potentials that we have really not a very clear indications. A lot of work has been going on, but obviously the nuclear membrane is a membrane structure as well. And if there's any damage to the membrane structure, you could result in the chromosome and DNA damage as well. Okay? Yes?
So if the hydroxyl radicals through the chain reaction can enter inside the nucleus, inside the nucleus also there are a lot of water molecules.
Yes.
They will make a chain of another hydroxyl radical.
Sure.
Then you should see the damage to the DNA also there.
Yes. Yes. And that's exactly what we will show you. All right. So one of the things, as mentioned, is that if you were to look at a biomarker for oxidative damage, and 8-hydroxydeoxyguanosine is one of those commonly used. Okay, so we have acquired a monoclonal antibodies from Dr. Regina Santella in the School of Public Health, and these are the control cells. These are the irradiated cells through the cytoplasms, and what we have done is to irradiate these, fix them, stain them with the antibodies, and quantify the staining intensities. All right, and so every single cell, as long as they breathe oxygen, they will produce free radical damage. They will have background inductions of 8-OHDG hydroxyguanosines. And if you were to look at these irradiated cells, the inductions will be much higher, as shown by this bar curve. So the control have some background inductions, and if you were to irradiate them with 8-alpha particles, you have a higher inductions. And to show that these generate as a result of inductions of reactive radical species, if you were to treat them concurrently with a oxygen quencher such as DMSO, you will be able to bring it down to the much lower level, indicating that the inductions of these 8-OHDG is related to the productions of the free radical species. All right? So, but this is just one of the many things that is happening in the cells that were targeted through the cytosol. And the other one is that if you were to look at these inductions of lipid peroxidations, and I mentioned that in that figures, lipid peroxidations is frequently generated as a result of the productions of free radicals. Any membranous structures, whether it's a nuclear membrane, mitochondrial membrane, or endoplasmic reticulums, they all be susceptible to oxidative lipid peroxidations. And so one of the approach that can be done is to measure the expressions of these 4-hydroxynonenal, which is a markers of lipid peroxidations. And if you look at the commercial probe from Molecular Probe and other companies, these antibodies are readily available. And so if you were to irradiate these cells with 8-alpha particles, okay, the one that give us the maximum response, and stain them with these antibodies for 4-HNE, you give you a fairly high induction level. And if you were to at the same time co-treat these with a chain blocker, which is one of those antioxidants, what we call a BHT, these will gradually bring down the inductions. Okay, so lipid peroxidations is one of the mechanisms of how the free radicals are being generated. I said one, there are many, many other pathways that could be involved as well. The other thing is that, as Dr. Matsumoto mentioned, nitric oxides is omnipresent in the cells. And so when nitric oxides are formed, they form as a result of the interactions with superoxides to form a very mildly reactive peroxynitrides. And so when these reactive nitrogen species are generated, they will bind to many of these protein products, and they will form these nitrotyrosines. Okay, and nitrotyrosines is also considered as a reliable markers for the productions of reactive nitrogen species. Again, if you were to use a commercially available antibodies, you can titrate and quantify the inductions of these peroxynitride anions in cells that are irradiated through the cytosol. All right, and this work has been published a year and a half ago, so I won't drill on it, apart to say that if you were to use a methyl arginine, which is a inhibitors of the enzymes that promote the formations of these radical species, you can certainly reduce the biological consequence of induced by these nitrotyrosine or the reactive nitrogen species. So there is a direct correlation with the productions of reactive radical species, whether it's oxygens or nitrogen species. All right, so it's all in the chart that we have showed you before. And if you were going back to these figures, formations of reactive oxygen species in the presence of nitric oxides with these enzymes, nitric oxide synthetase. And these methyl arginine is inhibitors of these enzymes. And if you block these, you won't have the productions of these peroxynitrides. And without these anions, your biological reactions will decrease. Okay? Is that clear? Any questions? Okay. So now, I have mentioned to you about these aspects, whether radiations induced targeted response. So the cytoplasmic reaction that we have shown you before was the first documented evidence of so-called the extranuclear response of ionizing radiations. So with the microbeam, and Dr. Matsumoto gave a very good introduction this morning already about the extracellular response of ionizing radiations. An extracellular response is what we have been talking about, the bystander or the non-targeted response. So in the cells, that population of cells, if only 1 or 2 or a certain percentage of the cells were irradiated, and these was best demonstrated using a microbeam in a series of published papers in the last 10 years, we were able to show with all of these endpoints, the cells that are not directly irradiated, but in the vicinity of cells that were, they were able to show a induction of biological response. All right? So with these, the next questions we wanted to ask. So far, everything about the non-targeted response is mediated through nuclear traversals. Okay? So if we irradiated the nucleus and the neighboring cells, we have demonstrated there has a bystander response. And obviously, now we were able to irradiate the cytosol. The question then, can cytoplasmic irradiations, which can result in the mutations in the nucleus of the same hit cells, would they have the propensity to induce a bystander response as well? All right? So it's a very simple logical extension of the first part of the studies. And so unfortunately, as we were doing that part of the studies quite a few years ago, our microbeam have some serious health issues. So we were not able to do the studies, and because it's a very competitive field, somebody stole our PNAS paper. And so this work was subsequently done by Kevin Price and Dr. Chunin Shao in Shanghai. So we have talked about these at meetings. We are on the way of getting this work done and publish it, and our machine broke down. And so now somebody else took it up, and what they have shown is exactly what we predicted. Okay? So if you were to look at the cells, he looks at human fibroblasts, and he irradiated these glioblastoma cells that are co-plated with human fibroblasts, and notice that these are the number of cytoplasmic traversal 1 particles and 10 particles with helium alpha particles, and he shows that if he were to irradiate these new glioblastoma cells with the alpha particles, he were able to show in the non-irradiated human fibroblasts the inductions of micronucleus. Okay, so this was the first demonstration that cytoplasmic irradiation with alpha particles can induce micronucleus in a non-hit cells. The first extracellular response, induction through cytoplasmic irradiations. And he also provided some evidence that if he were to use a nitric oxide inhibitors, okay, you must have heard this PTNO from Dr. Matsumoto, then he were able to reduce the inductions of this response. So this work was published by Kevin Price. So when our machine was finally get back in shape, we get back in the lab, this piece of data has never been published because we were trying to hold on to it and get to some higher end journals. But what it shows is that if you were to irradiate 100% of the cells with 8 alpha particles, this is what you have obtained, okay, based on the data that we have shown. So now, if we were to irradiate only 20%, and assuming that there was no crosstalk between the cells, this will be the actual yield that we have seen. Okay, and in actuality, when we irradiate 20% of the cells through the cytosol. When we collect the whole populations, this is the actual use that we have, and it's indicating we have a 4-fold increase. And all of these are what we projected to be through these crosstalk between cells through this bystander response. So now, moving onward, what is the signaling process? And we talk about this bystander response, but what is the significance? Okay, what does it do? Okay, why do we have to worry about it? So if you were to look at the signaling process, and Dr. Ping Narayanji, a couple of days ago, he gave a good presentations at this Microbeam workshop, and he already talked about some of the signaling process. It's actually very complicated, and these are work by Honing and Dr. Velomir in the groups, and a couple of review articles. In essence, when the cells are irradiated and receiving signals from the outside, they either produce interactions through surface ligands that stimulates the productions of these pan transcription factor, the NF-κB, and that indicated induce inductions of COX-2, cyclooxygenase 2, and or called the PGE2, and a range of cytokines and lymphocytes. And more importantly, when we irradiate through the cytosol, we were able to demonstrate that there is a potential damage to the mitochondria. Okay, so the question is that what is the biological response if we were to hit the mitochondrion? Now, with a microbeam of 1 micron, you can actually selectively irradiate a single mitochondrium. All right, so this was shown, work by my fellow Bo, who just just some very recent data. So if you were to look at a normal cells, and mitochondria, every single cells have several hundreds to over 1,000 mitochondria. And these arrange themselves about microtubular systems, and they give you a little elongated filamentous morphologies. All right, and so if you were to look at these under higher powered, give you very well organized, and if you were to irradiate themselves, these will show a more punctated structures, indicating there are some mitochondrial damage, and high power magnification will show it better. But with the mitochondrial morphological change, it also comes through with changes in mitochondrial functions. All right, so mitochondrial functions are controlled not only by the mitochondrial genome, but also partially by the nuclear genome as well. And so if you were to look at some of these oxidative respiratory proteins, such as cytochrome C oxidase, okay, and showing that these are the control in cells that are irradiated through the cytoplasm, they have a gradual decreases in cytochrome C oxidase functions. This is what we call complex 4. Okay, so mitochondria has quite a few complex. Cytochrome C is a complex 4. The other enzyme that we look at is SDH, succinate dehydrogenase. Succinate dehydrogenase is a little different from cytochrome C oxidase, because this genes, EMS proteins, is controlled partially by nuclear DNA as well. But nonetheless, when you look at the expressions as a function of time, there's a gradual decreases in enzyme activities, and gradually by 24 hours they will get back to their seemingly normal level. And so what these data suggest, when you target cytosol, there are biological response. Okay, and so these will then go on to show when we have these biological damage, they changes mitochondrial function. Mitochondrial functions are very important for the viability of the cells because they produce all the energies, all the ATPs. Okay, so when you have chronic mitochondrial damage, when you have increases in intracellular oxidants, what is going to happen? And you may result in these so-called genomic instabilities. Okay? Our current thinking is that when our body is constantly under stress, that's why your mom always tells you to drink vitamin C, okay? Have orange juice. And these then provided a source for removal of the stress and the oxidant stress, and resulted in genomic instabilities. So we wanted to ask, if we were to look at these cytoplasms, and these are the cluster of cells, if we were to irradiate through the nucleus or irradiate through the cytoplasm, will we be able to demonstrate in the progeny of the non-irradiated cells whether they will have genomic instability or not? Okay, a very important concept. We already know that if you were to irradiate the nucleus and you collected the progeny of the directly irradiated cells, we will have consequence, genomic instability. Now we are asking, what about the progeny of the bystander cells? Okay, not only through the nucleus, but also through the cytoplasms. And Brun Hu, who was a postdoctoral fellow in the Center for Radiological Research, working with Dr. Geer a few years ago, he did this work using the human hamster hybrid 11 cells, and Burung now has moved back to China at the Institute of Biophysics at the Chinese Academy of Science, and got a professorship as well. And so when he was here, he used these M-band and to look at the genomic instabilities of these human chromosome 11 among the hybrid cells. And the definition of genomic instabilities is to go back to these progenies, irradiate these red progenies through the cytoplasms, and collect all the yellow cells over here and follow them through a few passages, and to demonstrate that using M-band, the classic definition of chromosome genomic instability is that there has to have 3 of these metaphase populations with at least 5% of these score that shows genomic instabilities. So among these that he has looked at, this one fall into what we call as a genomic instability phenotypes. Okay, so these work currently under review. It was the first demonstration that targeted cytoplasmic irradiations, if we were to follow the progenies, were likely to result in genomic instability as well. And of course, genomic instability is extremely important in carcinogenesis because that's how cancer arrives. So to sum up what I have mentioned to you, the microbeam technology, of which you are here to learn about it, has empowered, changed how we thought about the radiobiological consequence of ionizing radiations. We talk about nucleus, now we have incidents of targeted cytoplasmic irradiations. So from the cytoplasmic damage, we have the non-targeted response, and from the tissue cells, we move on to tissues, and these provided a very clear-cut mechanistic insight of what actually happening to these cells, and in the long run, it provides better understanding of the biological response of low-dose radiations as a whole. Okay, so I think my time is up, and I'd like to see anyone has any questions.